OsteoBites is a weekly osteosarcoma webinar and podcast presented by MIB agents. Today we're speaking with Ryan Roberts, MD, PhD, a physician for the Division of Hematology and Oncology at Nationwide Children's Hospital. He's also a principal investigator at the Center for Childhood Cancer and Blood Diseases, Nationwide Children's, and a member of the Translational Therapeutics program at the James Comprehensive Cancer Center, Ohio State University. Our panelists today are osteo warriors and junior board members Ryan Kennington and Andrew Basaga, patient advocate Amanda Levine, and I'm your host, Anne Graham, president of MIB Agents. So welcome to OsteoBites, everybody. Grab your snack. I already ate mine, so I can't even share with you what it is. Let's go. Today, we are honored to be speaking with Dr. Ryan Roberts of Nationwide Children's Hospital. Since I visited Nationwide Children's Hospital, I think it was in 2019, and seeing the excellent work that's being undertaken at Nationwide and specifically by Dr. Roberts, I've been so excited about the work and really excited for updates. So here we are getting updated. Dr. Roberts shared his work with us as well at the Factor Conference. And then we have him here today on OsteoBites. We're an all osteo warrior panel today with MIB Agents junior board members, Ryan Kennington and Andrew Basaga, and of course, Amanda Levine, patient advocate, and also an osteo warrior. A little bit about MIB. We are a leading pediatric osteosarcoma nonprofit dedicated to making it better for a community of patients, caregivers, doctors, and researchers, with the goal of less toxic, more effective treatments and a cure for this aggressive bone cancer. Our mission is in our name, MIB, make it better for kids with osteosarcoma. We make it better through direct patient and family support, through education with our annual Factor Conference, OsteoBites, and our book, Osteosarcoma From Our Family to Yours, great resource. Was put together by our team at MIB and our scientific advisory team. And then we also do this through sharing and funding meaningful osteosarcoma specific research. If we can be of help to you, please let us know if you would like to join our shared mission to make it better. We also want to hear from you. Whatever it is you do it for kids with osteosarcoma. We need whatever magic you have to improve osteosarcoma. With that, Dr. Roberts, would you get us started by introducing yourself, please?
Thanks for having me, Anne. It's a pleasure to be here. I'm Dr. Roberts. I'm Ryan Roberts from Columbus, Ohio, and I'll tell you a little more about me in my talk, but I figured I'd just introduce you myself by sharing my OsteoBite of today, which is Junior Mint's Minis. So I love Junior Mints, but they made a mini now, which has improved the chocolate to mint ratio. So it's just right. And if you put it in your coffee mug that looks like a toilet bowl, people won't steal them from you. So my OsteoBite today is Junior Mint's Minis.
Oh my gosh, that is so awesome. I'd actually avoid your whole desk if I saw that.
Hi, I'm Amanda. Many of you know me from the Facebook groups of support. I was diagnosed with OS of my left distal femur and my left lung in 1987. I got married, I had 2 kids. Then in 2010, I was diagnosed with breast cancer. And my book, why I'm talking funny now, is because I was diagnosed with ALS in 2018. But even though I am failing, I will never fail my families that are fighting OS.
Hi, I'm Ryan. I was diagnosed with osteosarcoma back in 2014. I had a limb salvaging surgery, and unfortunately, a year after I finished treatment, I had a small relapse in my lungs. However, since that relapse, I've been NED ever since for almost like 4 years now. And I recently just graduated college from the University of Delaware with a degree in biology, and I am a member of the Schiffman Lab.
Hi, my name is Andrew. I was diagnosed with osteosarcoma about 3 years ago in my leg. I'm about 2.5 years cancer-free, and now I'm just trying to make it better for everybody else like me.
I thought I would share a little story with you guys today about what we've been doing in my lab here in Columbus. So I grew up in the beautiful mountains of Utah. Around 2002, in 2002, my wife and I got on a plane and took a trip out here to Ohio, to the land of the Buckeyes, so that I could go to medical school and graduate school. And I guess Ohio grew on us because we've been here ever since. I graduated medical school in 2010, and as I started to think about what I was going to do with my future, I met this guy. So this is Peter Houghton. Some of you may know him, and I joined his lab here at Nationwide Children's Hospital. And about the first 4 months of the time that I had in his lab was just trying to figure out what my question was going to be and how I could make a difference in the world. And I started out, just asking to join his lab, and he was happy to have me, but wanted to know what it is what the question was that I was going to ask. I went through a whole series of questions that he continually told me were just not good ideas. Or things that, like, there was already so many people studying that. Why didn't I choose something that had a bigger need for it? I thought about neuroblastoma, I thought about brain cancer and macrophages, going off of some of the things that I'd done during my PhD, but some things I just wasn't smart enough. Some things were just too hard. I thought about doing Ewing sarcoma, and he just kind of rolled his eyes and told me that Steve Lesnick would just crush me and Olivia Delattre would just destroy me like hors d'oeuvres. Interestingly, Steve Lesnick is now my boss. But after a while, I was having these experiences in the clinic, and I said, I keep seeing these patients with osteosarcoma. They get these lesions in their legs. They go through this brutal chemotherapy. We've got these fancy surgeries so that they can save their legs a lot of the time, which is cool. But, they finish therapy, they look fantastic. And then they come to clinic and get a scan, and this happens, right? And this is really where it hits the fan, right? They develop metastases. And once this happens, things get really complicated, right? Just none of the treatments we have really work for this. If we can't cut it out, we just feel impotent. There's just not a lot that we can do, and that just sucks. And so after 4 months of having him tell me what bad ideas I had, he finally said, you know what, that does suck, And I kind of started asking, why is it that it always goes to the lungs? Why doesn't it go anywhere else? What is it with that? And he said, I don't know, maybe you should figure that out. And I got really excited because I thought, if we could figure that out, then maybe we could do something. If we could figure out what is it about the lung that makes that such a special place, then we might be able to do something. And he thought that I was smart enough to do that, which was good. And he thought that, that was really a question that needed some attention, which was good because he kind of knows everything. And so that was the moment that my research love was born, and that's been my passion ever since, and that's all I've really studied. Although it should be maybe better stated that that's when my hatred was born for lung metastasis, and we've been working to eliminate that ever since. And so I want to tell you guys a story today about kind of some of the things that we've discovered over the last 4 or 5 years and how we're thinking about lung metastasis. I want to point out as we do this that osteosarcoma can be thought of as kind of a 3-compartment disease. You have the primary bone tumor that's inside the bones, and then you have the lung metastases that are inside the lungs, and then you have this other pool of osteosarcoma cells that are circulating, right? We call them circulating tumor cells. And so as we go through kind of how we think about lung metastasis, I want you to think about these 3 pools of tumor cells and how tumor cells get into and out of the blood as they go from the primary tumor into the blood and then into the lung metastasis. And I'm going to talk to you today about some of the processes that underlie metastasis. And I'm going to completely ignore everything that happens up until the tumor cell gets into the blood, because frankly, I don't care. Because when I see a patient, this part has already happened. And so I want to think about the things that I can do to target what's going on in my patients, right? And so dissemination occurs when the tumor cells spread from the blood into a distant tissue. And here we're showing the lung, right? Now, lots and lots of tumor cells do this. So tons of tumor cells will go from the primary tumor and end up in the lung. But what happens is that almost all of those tumor cells that get there die. They can't survive in the lung. The lung is not a happy place for them, right? It's totally different than the primary tumor, and all the support that they had around them within that primary tumor is gone. And so when they land in the lung, they just die. However, a very, very, very small percentage, for some reason, whether that be where they landed or some of the cells that are present or something that's just intrinsically different about a small group of tumor cells, lets them survive and then lets them expand into what is a clinically relevant metastasis. And so we've been asking questions about this process that we think can help us to understand it better, because almost nothing is understood about how a tumor goes from one step to the next to the next. And so one of those questions is, what is it that makes the lung different? Why don't we see tumor cells in why don't we see metastases in all of these other organs? And what is it about the places where the tumor cells grow, right? Are there certain cells within that niche, or are there certain signals within that niche that make that lung niche special? And are there certain parts of the lung that make that niche special? And then lastly, what is it about some of those tumor cells? Why do some survive and others don't? Is that just a matter of chance of where they land? Or is that an intrinsic property of those tumor cells? And I'll convince you today, there's two things that I want you to understand. And the first is that I want you to go away knowing that tumor cells depend on critical interactions that occur with the surrounding tissues. And I show you a few of those here, and we're going to talk about what these mean, so you don't have to memorize this. But I want you to understand that tumors don't develop alone. It takes a village to raise a tumor, just like it takes a village to raise a child. And the second thing that I want to convince you of is that metastases are more than just tumor cells. They are developing organs. So if you think about how development happens as a baby goes from a sperm and an egg to heart and brain and skin and bone, that's a process of development where different cells are brought in and certain cells change their phenotypes in order to become an organ or a tissue. And the same thing happens within a metastasis, right? And I'll show you just an example here. So here are several stains for different either behaviors or tissue types that are taken from a mouse at day 14 when the tumors are the smallest, when the lung metastases are small. So these are all lung tissues. Or at day 28, just 2 weeks later. And you can see that they're just different, right? You can see if you just look at the green cells here, there's none. And then all of a sudden there's a whole bunch. And this one we'll talk about, but surprisingly, there's no green no matter where we go here. These cells are excluded. And you'll see that these cells form almost all of the tumor in the beginning and then go away as the tumor grows. And you'll see there's other behaviors that seem to kind of rim the tumor and kind of coddle it as it grows. And so these interactions that occur with the lung tissues also seem to be important as the tumor grows. And what I want you to remember is that all of these interactions and all of these changes are important as a metastasis goes from a single cell to a developed organ that is a clinically relevant metastasis. And one point that I want to make is this. While a lot of attention has been given to cells that infiltrate the lung when tumors develop, we have kind of learned to focus our attention on the actual lung cells themselves, which have been largely ignored. They've kind of been assumed to be kind of inert in this interaction. But what I have here is a video of a tiny tumor. It's about a 12-cell tumor, and the tumor is the red part of this image. And the green cells are all epithelial cells, so the structural cells of the lung. And you can see that they are just all over the tumor cells when these tumor cells are tiny. And we think that when the metastases are tiny, we think that when these metastases are tiny, that's the time when we need to intervene, because that's probably when they're the most vulnerable. And so we focus a lot of our attention on these early stages. So we showed several years ago that IL-6 and CXCL8, these are two inflammatory cytokines, so soluble signals that cells produce that cause inflammation. And we showed a long time ago that these two signals were necessary for metastases to form. And so this was an experiment where we just put tumor cells into mice, and we blocked IL-6 and CXCL8 signals using two drugs. And when we did that, this is a survival curve, and you can see that we started out at time 0, we treated for 42 days, and over time, all the mice that didn't get treated began to die of lung metastasis until all of them were dead. But about 85% of the mice that got this treatment were long-term survivors. Even though we'd stopped treatment, they go on living, and there's no evidence of metastasis. We had no idea why this happens, but we knew that it happened, and it happened reproducibly in lots of different tumor models. And so a lot of what we've been doing has been trying to figure out why, and why does this happen, and what is it that these do, because this is unfortunately not a clinically translatable intervention for a number of reasons. And so one of the things that we did is we tried to figure out who's producing the IL-6 and CXCL8, these inflammatory molecules. And it turns out that we thought it was the lung at first, but it turns out it's not the lung, it's actually the tumor cells themselves that produce this. And so once we figured that out, we were really trying hard to figure out what signals from the lung cause the tumor cells to produce these inflammatory cytokines. And this is just some really elegant work that was led in large parts by Marin Kam, who is a fantastic molecular biologist, in my lab. And we were able to show that IL-1 is produced by the lung cells, and there's receptors for IL-1 expressed on the tumor cells, and that interaction drives a whole panel of genes that are associated with this phenotype that we see in the cells that survive in the lung. And so we think that IL-1 is the driver of this interaction, and we can kind of prove that here with some experiments done by John Hinckley, where if we take osteosarcoma cells alone, they don't produce a lot of IL-6. If we take bronchial epithelial cells, so those structural cells of the lung, and look at those alone, they don't produce a lot of IL-6. But when we put them together in co-culture, now all of a sudden we just crank out boatloads of these inflammatory cytokines. And interestingly, if we block IL-1, it goes away. Or if we block IL-6, it goes away, which suggests that there's a paracrine loop where each cell is kind of amplifying the signal of the other to create this inflammatory reaction. And so you can see here the stains of the IL-1 that I showed you earlier. You can see where the tumor cells interact with the epithelial cells. The epithelial cells are producing a bunch of this IL-1, and the osteosarcoma cells in return are producing IL-6. And that interaction just feeds forward to kind of crank up the volume. So we've kind of said, okay, but what does that mean, right? And what does that do? Well, I told you that these epithelial cells are important. So what does the tumor do to the lung epithelium, to these structural cells of the lung? Well, we've learned that answer recently, and the answer is cool biologically, but strange in a way, and that is that the tumors that go into the lung actually produce a wound that can't heal. So those signals, those inflammatory signals within the lung, cause those epithelial cells to change their phenotype and to prime themselves for wound healing. And so they start to proliferate, and they create a whole bunch of these, they're called transitional epithelial cells, that are ready to go in and cause this wound to heal. But that doesn't happen because the cancer is still there. And so this becomes a chronic wound with an accumulation of these epithelial cells. And one of the things that these epithelial cells do is that they produce a bunch of growth factors and a bunch of cytokines that actually feed the tumor. So in this way, the tumor creates, it kind of fluffs the pillow of the surrounding environment, right? To make it a really nice place where those tumor cells can grow. And James Renicky in my lab has done some just incredible work kind of showing how that happens and how we might actually be able to intervene on that. And you can see this here in kind of in a zoomed out picture. So these are pictures of a couple of different models of osteosarcoma metastases. And what you can see is that there is this keratin 8. So everywhere there's green, that's these transitional epithelial cells. And you can see the tumors here. Here's one here, and you can see these tiny tumors here up on the F420 side. And everywhere that there's a tumor, you get all these kind of wound healing epithelial cells, and they become just a massive part of what's inside the tumor. And so how does this affect the growth of a metastasis? As I mentioned before, that this is kind of like fluffing the pillow. And so we've actually shown that this seems to occur, that process seems to occur. I want to show you kind of a cool experiment that we did. So what we did is we took osteosarcoma cells and we split them in half. And half of the tumor cells we labeled with a green fluorescent marker, and half the tumor cells we labeled with a red fluorescent marker. And we took the green cells and we put them into the mice, and then we waited 10 days. And we waited 10 days because that's about how long it takes for almost all of the cells to die off. And what you're left with is just the ones that's going to survive. And so as soon as we got to that point, we then took the red cells and put them into the mice. And then we waited 10 days and we harvested the lungs. So what do you think would happen, right? Well, you would predict that when we harvest those lungs, we'll see a whole bunch of great big green tumors because they've been in there for twice as long, right? And we might see some little red tumors as well. But that's not what we find. What we find are tiny little foci of green cells surrounded by red cells, right? And so this suggests that those green cells were remodeling this environment to be more conducive to the growth of the tumor cells that would come in later. They're kind of like paving the way, right? And we're going to talk a little more about how that happens and what those tumor cells are. And I just want to thank Melissa Salmons, one of my animal gurus who helped do a lot of these experiments. She handles a lot of our mouse colonies. And so James Renicky in my lab made a really interesting observation that kind of explained why all of this might happen, right? How it is that that some cells might die and others live, and how it is that they might kind of fluff that pillow within the lung. And the first observation he had is we'd kind of shown that when we exposed these tumor cells to IL-1, as I showed you earlier, they produce all this IL-6. But when he did just a stain where you could look cell by cell to see who's producing the IL-6, it was actually just a really, really small number of the cells. So just a few of the tumor cells were producing all of the IL-6 and all of the IL-8. Which was just bizarre. We didn't understand why this was happening. And we've come to understand since then that these really represent kind of a population of cells that are there within the tumor, and the IL-1 just kind of unmasks what's under the hood, right? And so they might look all the same on the surface a little bit, but once you start seeing how they react to different stimuli, you can see it. And so this is a single cell analysis. I won't get into details, but the closer that two dots are, each dot is a cell, and the closer that two dots are, the more alike they are. And you can see that when we culture osteosarcoma cells here on the right with epithelial cells on the left, there's this little population that emerges, kind of comes away from the tumor cell. And these are those cells that produce IL-6 and IL-8, and they're P21 positive. And that's important because P21 is actually a marker of senescence. And P21 positive cells typically don't proliferate, right? So that was really confusing to us, and we didn't really understand why. Why is it that this population of cells that can't even proliferate would be so important for the formation of metastasis, right? You'd think that metastases are cells that are just growing out of control, but that turns out to be not the case. And I showed you a little bit of this data earlier. We showed that in the teeny tiniest of metastases, essentially all of the tumors have this P21 positive, IL-6 producing, CXCL8 producing phenotype. And we've started to call these anchor cells for that reason, because they're the first cells in, and they're the ones that survive that initial attrition. And they are the ones that fluff the pillow for the cells that come later that then grow to become the actual bulk of the metastasis. And so we've come to appreciate this as kind of a cooperative event where not all the tumor cells are the same, they're different. And just like a developing organ, right? I told you earlier to think of this as a developing organ. The cells can divide the work that needs to be done to create this tumor and then cooperate to make it happen. And I'll just very quickly show you some data from one of my graduate students, Sanjana Rajan, who has kind of shown that this seems to be an intrinsic property of osteosarcoma cells. And this is a paper that we just released online just a few days ago, so I'm not going to belabor it. But I'll just tell you that within every culture of osteosarcoma cells, whether that's something that came straight from a patient, whether that's something that's been cultured for months or years or decades in plastic, or whether that's some of our PDX models that have been in mice, they always retain populations of cells that have different phenotypes. And this is preserved even when there's clonal selection, as happens like with a metastatic event. And so there seems to be some intrinsic property, and in the interest of time, I'll skip over this a little bit, but there's an intrinsic property to these cells that these are not just monotonous cells. They're different, and they distribute their function. And if you isolate one type of cell from that group, it will again create a different set of cells that has different functions. And so we can talk more about that later if you want to, but it's some interesting data. And so kind of a linchpin in this analysis was to show that actually these tumor cells interact with each other. And here's an example of that where we showed that if we just use conditioned media, so that's fluid taken off of cells in culture, and we put that in the bottom of a transwell, and then we put osteosarcoma cells on the top, we showed that they're attracted to the signal that was produced by the tumor cells. And if we block IL-6 and CXCL8, as you see, as we go to the right here, so here's blocking IL-6, here's blocking IL-8, and here's blocking both of them, you can see we lose that chemoattractant motion. So we lose the motion in response to the factors that are produced by the tumor cells. And so we think that this signal that's produced in the lung, where they're kind of feeding forward and producing all these cytokines, also draws new sarcoma cells into that metastasis. I showed you this picture before, right, where the green tumor foci were recruiting the red tumor foci. We think that's probably what's happening, is that they're kind of saying, hey, come here, I've got a nice place for you to live, right? I fluffed the pillow and now you can grow here. And there's some significant implications for this, and I'll just point to one of them, and that is that I told you that those anchor cells don't proliferate, right? Well, if they don't proliferate, then they won't respond to chemotherapy, right? And so if we have these anchor cells embedded within each of the metastases that occurs within a lung, well, then using chemotherapy won't kill them all off. And I'm sure you guys all kind of get that intuitively, right? And you can show here if we let a tumor cell grow till it's a decent sized metastasis, and then we treat it with chemotherapy, you can see these same P21 positive cells that were there when it first started are present after all the other cells die. And so our current work in the lab is working to see if there's a way that we can target these anchor cells specifically, right? And if we can do that, then we might be able to not just prevent metastases from occurring, but we might be able to use it in combination therapies that would make these lesions treatable, right? Because they're so hard to treat, as I mentioned before. And I'll just say that that's what we're working on. And there's some things that I think we're kind of hopeful for, and we're working through those now. But it seems that if you can find things that target the tumor cells, and this is a really cool assay that we do where we kind of grow metastases on lung cells in a dish. And if you can get rid of those P21 positive tumor cells, the metastases just blow apart. They can't hold together anymore. And so we think that this might be kind of a linchpin to be able to get at these metastatic lesions. And so with that, I'll thank all the people in my lab who helped make this possible. And I'm happy to take any questions.
Okay, my brain is full.
Sorry, was that too much?
No. I want to make sure I'm following. And I guess my question on the P21 is, and I mean, our understanding of osteosarcoma, or at least my understanding, and what seems to be the a result of a lot of studying a lot of osteosarcoma tumor cells, is that osteosarcoma is so different in each person, right? My osteosarcoma doesn't look like Amanda's or Ryan's or Andrew's or pretty much anybody else's, which is why why it's such a difficult puzzle to solve, right? Mm-hmm. So the P21 is that expressed in all osteosarcoma?
So
at a target in all osteosarcoma?
You ask complicated questions, Anne. But let me, so a hallmark feature of nearly all osteosarcomas is loss of P53.
Right.
And so that's something that we know is common to almost all osteosarcomas. One of the primary targets of P53 is P21. Okay, and so P21 is usually the executor of the P53 signal that says stop dividing. Okay, and so while you are 100% correct, and I didn't get into it, but we actually showed exactly what you had said in the paper that Sanjana did that we just released online. What she showed is that two tumor cells are way different from each other than they are from their own lung metastasis. So two lung metastases from two different patients are way more different than each of those is to their primary tumor, for instance. Okay. However, you can show that as a tumor goes from a primary tumor to a metastasis, there is a certain set of events that appears to be critical for navigating that process. Okay, and so while yes, you are right that that one osteosarcoma cell is different from another, one lung is not that much different from another, right? And so some of these processes that drive the interactions between the tumor and the lungs might be more alike than different, and might be far more alike from one tumor to the next than the actual biology that's going on inside the tumor. Does that answer your question?
Yeah. Yeah, I think so. And then I guess the other question, and everybody, please ask your questions because I'm sure they're going to be more interesting and intelligent than mine. So please ask away. Just put it in Q&A or in chat, and we'll get to them as soon as possible. I guess my other question is, what role does the tumor microenvironment play in this? Because it's like the soil, right? Is that the same thing that you're talking about with fluffing the pillow, where the sort of the soil being the lungs, and this just creates a great place for everything to grow because the soil is amenable to these seeds coming in and taking root?
Well, you're spot on, Anne. I mean, this is, so we talk a lot about the seed in the soil, right? And then there has to be a good match for a tumor to grow. But we don't know what it is about the soil that makes it favorable to that seed, to the osteosarcoma cell, right? So that's kind of what we're studying here. And what I've shown you are mechanisms that make that soil fertile, right? And so these are interactions that specifically occur between that seed, the osteosarcoma cell, and that soil, the lung, that, you know, I said fluff the pillow, but you could say fertilize the soil, right? Yeah. To make that a place where those tumor cells can grow. As I mentioned, almost all of the tumor cells that get to the lung will die, right? And it's just, there's a few chance interactions that occur that allow a cell to anchor within the lung, right, and survive. And so that's kind of what we've been asking, is what is it that lets that happen? And these interactions that occur between the tumor cells and the lung cells, we think are critical to making that process go forward.
Okay. So at what point, because one set of slides was showing like 2 weeks on, it goes from like this tiny, you know, sort of controllable situation, seed in the soil, where that seed is just a little tiny thing. And then in 2 weeks, it seems to go kind of out of control. So is this work working toward kind of a preventive, you know, kind of a preventive measure before things get out of control? And if so, is that preventive measure possible to just give to everybody who's undergoing osteosarcoma treatment? How is
You've been talking to Damon Reed. So I would say yes to all of the above. As we approach this question, my intent was exactly what you said, right? Yeah. Was let's take these patients who are NED at the end of therapy, and let's figure out what it is about those cells that are still alive. Let's find them and let's kill them, right? Yeah. Or that there's some data, especially in breast cancer, that says maybe what we should do is to try to maintain dormancy, right? To keep them like that. As we've gone through and we've kind of discovered this, it's kind of also given us an insight into why these metastases might be resistant to therapy. And so the same mechanism might actually mediate both survival in the face of the stresses from going to new environment, and survival in the face of chemotherapy for totally different reasons, right? It might just be coincidentally that same process drives both. If that's true, and so far it seems to be, then the same treatment might actually meet both objectives, right? You might be able to use the same treatment both for keeping metastasis from occurring and for making existing metastases treatable.
Which would be fabulous.
Which would be fabulous, right? But well, we've got studies going on right now. We just got a great grant from the Osteosarcoma Institute to study exactly that question. And so we're modeling that in a bunch of our animals and in dogs. I will say in the dog studies, there's some early, really encouraging data.
Yeah.
And I have to say, I feel like right now in osteosarcoma, we finally have this team and this cohesiveness within the entire community, with everybody working together. Places like Osteosarcoma Institute and MIB and people who are really meaningfully funding osteosarcoma research. I am so hopeful that we're changing the trajectory of this disease. And of course it's not soon enough for too many families and for too many kids, even who are in treatment now. It's not fast enough. And I know you know that better than anybody because you face the patients every single day. But I feel like I did when I was at Nationwide Children's Hospital, you know, talking to you and your team and just going, okay, this is really great work. And I think there's hope here.
You know, I have to agree with you. We published a review maybe a year ago that was titled "Accelerating Progress Makes for a Hopeful Future."
Mm-hmm.
And I just took on the role as the head of the Osteosarcoma Biology Committee for COG, which is a lot of work, but I did it because I think we really are at a time of real opportunity.
Yeah.
There's a lot going on right now, and it does make me really hopeful for that we can do something, that this, we haven't seen improvement for 40 years. It's about friggin' time, right?
Exactly. Yeah. True story. Speaking of hope and great things happening, Dr. Kurt Weiss has joined us, and he has a question for you.
You're more a soil guy than a seed guy, and I've been trying to compare primary tumors and Mets from the same patient with RNA-seq, but you're comparing. You're taking a deep dive into the microenvironment. So, like, I'm the mint and you're the chocolate, or vice versa.
Yeah.
So that it sounds like maybe we should team up. And I'm so excited to hear that you're taking advantage of the awesome vet school there at Ohio State and doing the canine model of osteosarcoma. I think it's really exciting.
Yeah, thanks, man. You know, we made an attempt to connect a while back, and then I think we both got busy. But we should do it again, because I think you're exactly right. There's a lot of ways that I think you and I could work together.
Well, it's beautiful
and I
work, Ryan.
Completely agree.
It's beautiful work, and congratulations, you're killing it.
Thanks. You know, Rick Lamp brings up a really good question. He asked if anyone was evaluating the IL-1 agent Canakinumab for lung metastasis and osteosarcoma. So Canakinumab is an antibody treatment that's used for rheumatoid arthritis.
Yeah, it's the IL receptor antagonist protein.
It actually targets IL-1 beta. So the interesting thing about Canakinumab is that it specifically binds IL-1 beta.
Okay.
However, there was just a big clinical trial testing its efficacy in preventing atherosclerosis in heart disease. And while it was a total failure at preventing heart disease, that CANTOS trial showed that it prevented the development of lung cancer. So that was kind of like the surprising finding that no one had expected, and I thought it was really intriguing, because
isn't that fascinating?
Maybe there's something to that. In osteosarcoma, however, Canakinumab won't work, because most of the activity that we see, I know I showed you IL-1 beta today, but most of the activity we see is actually IL-1 alpha driven.
Right.
And so Canakinumab won't work. However, some of the other agents might, and we're testing those now.
Well, fantastic. Congratulations.
Yeah, thanks, man.
Other than by chance, do you think there are any inherent characteristics behind the number of relapse sites, or even behind patients having multiple relapses in the lungs?
I hope to get to a point where I can answer that question. Okay. I think right now we can speculate as to that there might be some things that would make one patient's tumor more likely to form metastasis than another, but we just have to do the studies to figure that out. Right now we have the tools and the concepts that would let us ask that question, but I haven't been able I don't have the data to be able to tell
Okay.
you.
No, yeah, that's fine.
Tell
was
you
trying to know
truth.
more.
I can tell you opinion, but I can't tell you truth. Good question, though.
At the end, I think it was after 4 weeks, you used 2 chemos, cisplatin and it looked like Cytoxan. What made you choose those 2, especially Cytoxan isn't really used anymore? I had it, but
Well, when we're working with mice, we have to use drugs that work in mice. And so we do use ifosfamide a lot in patients, right? So ifosfamide kind of supplanted the use of cyclophosphamide in humans, but in mice, cyclophosphamide is way easier to use just because of some specific toxicities in mice. And so it's a model of kind of relapse therapy for mice when we use Cytoxan. Good question, though. You picked up on that. Surprise. Good job.
I have 4 chemos that they don't use anymore.
Yeah.
Me and everyone that was sick with it all surviving, and that's 33 years. So it drives me.
Did you have bleomycin?
And
Yeah.
And
you know, it's interesting because bleomycin actually induces that phenotype in the lung cells if you get high enough doses, and so we're actually looking into that too as a model.
Just as a side funny thing, Amanda and I, we call ourselves the Myers sisters. We're both treated by Dr. Myers.
Yeah.
Like, what, 30 years apart? 40 years apart?
Yeah.
The thing is, I was sick, but it was 5 of us, all within 2 years of age and 2 months of diagnosis, and we're still together this day, but we had different chemos.
How do TKIs affect or interrupt the process?
Are you talking like so like regorafenib and cabozantinib and these things have kind of become the, I don't want to say the rage, but have been like a super useful tool that has developed, especially for late stage disease. The most appropriate answer is we don't know. Okay, the hard part about studying what we call the multi-TKIs, the MTKIs, like cabozantinib, pazopanib, regorafenib are the 3 big ones that we use now. The hard part is that they target lots of different pathways kind of simultaneously and all a little bit, generally. And we haven't done enough research to know what makes one tumor respond over another to those drugs. We have some ideas, like for instance, they all target VEGF, and we have some tumors that are really high on VEGF. Whether that means they're dependent on VEGF or not, we don't really know. And so that's at this point in time an unanswerable question, but is something that is being investigated actively. Rich Gorlick has a bunch of work in that area, as does Alejandro Sweet Cordero. And so we may have some more data on that in the coming years, but we don't know right now.
All right.
Good question, though. Yeah.
So if you remove the initial mets, do the lung epithelial remain as fertile pillar fluffers such that any other CTC might have an ever increasing potential of surviving?
That's an excellent question, relevant to kind of the treatment that I propose. And I can answer that question in 2 weeks. But I don't have the answer yet. We're doing that experiment right now where we're actually kind of doing that red-green experiment, except we kill off all the tumor cells before we inject the second round. I would predict that for at least a time, that pillow remains fluffed, for lack of a better term. And so it doesn't go on forever, but I don't know how long that will last. But that's an excellent question. That's actually something we're concerned about.
We need to have you on again in sort of the new year to get another update. Right? So will you come back on in another?
Sure. I'm happy to talk to you guys anytime you want.
Okay, well, listen.
Great group. I'm a big fan of your organization and of this format. So
Oh, good.
Always happy to talk.
I'm so pleased. Thank you. That's that means a lot to us. You might change your mind after we ask you our burning questions.
I doubt it. I doubt it. It would take a lot.
Well, let's see. Let's go with our burning.
Let's give it a go. Let's give it a go.
Let's go, Ryan Kennington.
All right. So the first one is, what other profession would you like to attempt?
A high school science teacher. If I wasn't a doctor, that's what I'd be.
Yeah, you'd be good at
Mm-hmm.
that.
What was your first job?
My first job was the cleanup boy in a bakery. I scraped dried glaze off of every surface imaginable, and I couldn't eat a glazed donut for like 10 years after I had that job.
Your favorite snack, which I think we might already know?
Junior Mints, baby.
Okay, wait, did you see that Seinfeld episode with the Junior Mints?
I did. Yep. It's a fantastic episode.
It's one of the best episodes.
Yep.
Right. Sorry, go ahead.
What do you hope for?
Well, I hope that this work is meaningful sometime and that it helps patients, right? That one should be obvious.
What superpower would you choose?
Oh, easy. I'd clone myself. I want, Jack Jack, where all of a sudden there's like 20 Jack Jacks. I want to be able to do that so I can get more done. Because everything's always waiting for me to do it.
What inspired you to become a researcher?
Just my love of science. I just think science is cool, right? It's so cool. Like, I talk to my grad students. This might sound weird, but like what turns you on, right? And when I get up in the morning and know that I get to come to the lab and play with a set of data that no one's ever played with before, and that I might learn something that no one's ever known before, that gets me going. I love that. That gets me in the shower and makes me excited for my day.
What inspires you now?
Oh, my patients. It's both. I might say motivates me more, although inspires me too. But I hate not having better answers for them, and I feel some personal responsibility to change that.
What's your favorite word?
My favorite word. My favorite word is done. I like work that is done and not still on my plate.
What do you need to accomplish your work?
To clone myself. and resource. I mean, I think all the researchers will answer the same question to that one, right? It takes a lot of money to do good work, right? And good people are expensive, and good techniques are becoming increasingly expensive, although also more powerful at the same time. And so all of us as a community, I'll just say, are humbled at the sacred opportunities that you all give us to be able to do our work. And I hope that we one day give back to the community in a way that you feel like it was all worth it.
Can I just?
And then last
We're
question.
trying.
There's a reason we asked that question, and it is because until you know this, you don't know this, that the researchers are not funded entirely by their institution or entirely by the NIH, and that so much of the work that gets done in a lab comes from organizations like St. Baldrick's and Osteosarcoma Institute and MIB agents, and it's really important to, if this is important to you, which thank God it's important to a lot of people who are able to help fund research, we have to continue to fund research. It is our moral imperative to fund the important work that's happening to make it better for our kids. Like, otherwise we have bereaved families, and we have kids who are suffering terribly and then dying. So the reason we ask that question is to inform.
Yeah, you know, I've got a bunch of friends that are like lawyers and IT professionals and things like that. And when we sit down and talk, and they find out that like I have to fund 80% of my salary so that I still have a job out of my grants, they're just flabbergasted. They can't understand how that would work. But that's the case for most of us that do research, right? Is that we have to fund not only our own research, but our own jobs, right, in order to keep going. So most of our time is spent writing grants and things to try to keep things going.
All right, and then last question, would you rather repeat high school or med school?
High school. I loved high school. High school was so much fun. I would go back and do it again if I could. I have so many good friends, and we did so much mischief that was just fun. Not illegal, but fun.
You're the only person who's answered that.
I couldn't get away with it now, though.
Yeah, it's good stuff.
Yeah.
Okay, we're super over time, so I'll wrap this up by asking everybody to please go to our website and participate in Shopping for Hope. It is extended through this Sunday. It helps fund research and MIB Agents program. This program has exceeded our expectations on so many levels, including the fact that nearly every shop that's participating in our Shopping for Hope program they are connected intimately to osteosarcoma. For example, Charlotte Murdoff, who's a regular panelist on OsteoBites, who is in active treatment, makes these beautiful, sews these beautiful hoodies and sells them with 100% of the funds raised going to osteosarcoma research. Amazing. Danielle Mock's son, Carter, passed away from osteosarcoma. When he was in treatment, the two of them would take bicycle parts and make jewelry out of them. Carter passed away 2 years ago. His mom continues to make these beautiful earrings and necklaces. She's one of our shops. Sensi, Pampered Chef, Zaya Activewear, Useborn Books, Pure Haven Toxic Free Cleaners, Lip Sense, Color Street Nails, Damsel in Defense, and Kendra Scott Jewelry, which I might have gotten some gifts that I think I've got to buy double in because they're so cute and I want to keep them. So now I've got to buy more for my friends and stuff. Anyway, all such good stuff, and it all goes for such a good cause. So I pretty much did all of my shopping on Shopping for Hope. So please, 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 please shop for hope with us. This is it, agents, our last OsteoBites of the year and the end of our first season. What started as a way to keep in touch with researchers and their work in April because of COVID has become a thing. We now have a comprehensive library of great osteosarcoma research and researchers on YouTube, also on our website and podcasts. And we have, as it turns out, only scratched the surface. We have so many great guests coming up in January and February. We're starting to book into March. So stay tuned after the new year and we'll get started again. Full schedule coming up. Thank you for joining us today. And of course, thanks to our guest, Dr. Roberts, for your work and for sharing your time and your expertise with us. It matters so much to us. And also thanks to our panelists, junior board members, Ryan Kennington and Andrew Basaga, and Amanda Levine, patient advocate, osteo warrior, and my friend, and I love her, and I love all you guys. Thank you. Thank you, thank you. So we'll see you again.
Thank you for having me.
Holidays.
Thanks for all you guys do.
Thank you.
Look what came.
What did you get? Oh my gosh. Yes, count me in.
I know, I saw you got yours, so this just came in the mail.
Okay, you guys, if you have not signed up for Count Me In and you or your child has had osteosarcoma, osproject.org. This is a way to meaningfully participate in osteosarcoma research. This information really, really matters, and it's free. It's free to all of us, and it's a way to make it better. Really, really, osproject.org. Okay, thanks again. Thanks again, everybody. Happy holidays.